This section explains what biocompatibility testing actually measures. Biocompatibility is a property of the material in a specified use, never of the material on its own. Williams' definition captures this: the ability of a material to perform with an appropriate host response in a specific application. That phrasing folds host, site, and duration into what is being tested, rather than leaving them in the background. So ISO 10993, Part 1, tests nothing itself. It is the router. The pairing of the device's contact site with its exposure duration indexes which battery of tests runs. That battery covers cytotoxicity, genotoxicity, blood interaction, implantation, and systemic toxicity. It is graded, and it escalates in vitro before in vivo before clinical, with cheap screens gating the costly ones. The criterion is appropriateness rather than inertness. A device can pass every toxicity test and still fail. A bone plate that depresses the very bone it fixes is exactly that case. This section explains why biocompatibility is a relation the material enters rather than a fixed property it carries. You never test a material in the abstract. The ISO 10993 framework makes this operational. You locate the device on a grid. One axis is how it touches the body, surface, blood, communicating, or implanted. The other axis is how long it stays, transient, prolonged, or permanent. The cell where those two coordinates meet hands you the battery of tests owed. Intimacy and duration are the two knobs. Every required assay is a function of them. Read off a new device's coordinates and you can predict the tests it needs. The same steel is safe enough as a scalpel and under full scrutiny as a heart valve. This section explains why Part 2 of the ISO 1993 battery stands apart from every other part. Every other part of the ISO 10993 battery answers what the device does to the body: cytotoxicity, genotoxicity, blood interaction, implantation effects. Part 2 alone inverts the axis. It regulates what the testing does to the animal carrying the assay. This is the 3R constraint: replace, reduce, refine, bound into the standard itself. Because of it, generating biocompatibility evidence is priced against animal cost rather than treated as free. The chapter only names it one row in Table 1.4. But the pattern generalizes. Wherever a measurement consumes a living proxy, expect the welfare clause folded into the protocol itself, present from the design of the assay rather than appended once the assay is done. This section explains why one part of the ISO 10993 battery does something none of the others do. Every other part names something to measure in the material: genotoxicity, cytotoxicity, degradation products, blood interaction. Part 2 measures nothing. It is the single member that constrains the tester rather than the tested. Wherever an assay reaches for a live animal, Part 2 gates that reach through the 3 R's: replacement, reduction, refinement. So the biocompatibility regime carries one orthogonal axis. It is a material property series with a conduct constraint threaded through every test that spills into a living body. Grasp the shape, and you predict it elsewhere. Any measurement standard whose procedures touch living subjects grows exactly one non-measuring ethical side clause. The assay battery regulates its own hand. This section explains the tier of harm that these 3 tests isolate. A material can be harmless to the cell it touches, yet ruin the information that cell carries forward. That is what genotoxicity, carcinogenicity, and reproductive toxicity are built to catch. Cytotoxicity asks whether a cell dies now. These 3 ask something further, whether the damage propagates through cell divisions into tumors and through the germline into offspring. The organizing primitive is depth of harm, read as time scale. The deeper the target, the longer the injury outlives the exposure. Bundled under ISO 10993 Part 3. They sit at the far end of the biocompatibility battery because their endpoint is not a wound, but a lineage. That is why permanent contact implants trigger them, and a limited contact device need not. This section explains the tier of toxicology testing that looks past whether a material kills a cell and asks instead whether it corrupts the information that cell carries forward. Cytotoxicity asks whether a material kills the cell in front of you. This tier asks the harder question, whether it corrupts the information that cell carries forward. Genotoxicity tests damage to the genome itself. Point mutations, chromosome breakage, the errors that outlive the cell that made them. Reproductive toxicity extends the same logic across whole organisms, asking whether exposure degrades fertility or the developing offspring. The unifying primitive is latency. These are the failure modes. Silent in an acute readout, surfacing only down a lineage, a daughter cell, a generation. You assay them separately, precisely because the exposed body's own survival is a poor proxy for the integrity of its code. This section explains how ISO 10993, Part 4, selects the tests for a device that contacts blood. Blood does not fail one way. A surface meeting, it can seed a clot, consume platelets, shear red cells open, or trip the complement cascade. These are independent channels, and no single number covers them. So the standard refuses a universal assay. It indexes the required tests by how the device meets blood, where along the flow path, and for how long. Then it draws the matching subset from 5 categories: thrombosis, coagulation, platelets, hematology, and immunology. The move that makes this predictive is that a test battery is a function of interaction mode times exposure. The material itself does not set it. You predict the next case by asking where blood touches and for how long. A transient catheter and a permanent valve, though both blood contacting, share almost none of their tests. This section explains why blood compatibility is not a single test, but a partition of the failure space. Blood has no single compatibility axis. It is a bundle of independent, self-amplifying cascades, clotting, platelet activation, complement, and the circulating cell counts themselves, each one tripped by a different surface cue. So the chapter does not ask whether a material is safe in blood. It partitions the question by cascade. One test battery for each material is safe in blood. It partitions the question by cascade. One test batemium. The generative move is to partition the failure space by mechanism. Enumerate the distinct ways blood destroys itself on contact, then assign a probe to each, because a surface can be inert to coagulation yet still ignite complement. Compatibility is a checklist over orthogonal catastrophes rather than a property of the material. The standard's 5 categories are exactly that partition. This section explains how cytotoxicity is screened in vitro before any animal ever sees the material. Toxicity gets screened first by interposing a confluent lawn of standardized fibroblasts. the L929 line and its cousins, between the device and the organism, then challenging it two ways. The first way is direct. The material sits on the cells. The second is indirect. Only its extract does. The leachables pulled out under fixed conditions, a set area, to volume ratio, 37 degrees Celsius, at least 72 hours, then pipetted onto the monolayer. Read the damage as morphology and growth loss against paired positive and negative controls. This is the cheapest first tier gate of the ISO 10993 battery, the one test even the simplest catheter cannot skip. The generative move is this. Cytotoxicity is dose delivered to a proxy tissue. So every knob, extract ratio, time, cell line tunes the delivered dose. This section explains how the living body reduces to a small set of building blocks. The body comes down to 4 monomer families: amino acids, nucleotides, sugars, and lipids. Each family polymerizes into one macromolecule class, and those classes stack upward into cells, then tissues, then organs, then organs, then organs, then organs, then organs, then organs. A small alphabet generates the whole. This is the compositional hierarchy. Primitive, the same move as a grammar spinning every sentence from a finite lexicon. The turn that makes this matter is that these exact molecules are what an implant first touches. The host is itself built from polymers, and several of them double as biomaterials. That is the boundary worth predicting from. To engineer what goes into the body, you must read the body as a material, the same chemistry standing on both sides of the interface. This section explains how the test for local effects after implantation reads biocompatibility. Biocompatibility is never read absolutely. It is read differentially. The tissue reaction a material evokes where it sits, scored against a known good control implanted in parallel. ISO 10993, Part 6, owns the place, bound half of that reading. And place the device in a representative bed, subcutaneous, muscle, or bone. Wait. Then examine the interface it built histologically, looking for inflammation, fibrous encapsulation, and necrosis. The generative move is the partition. The body's response splits by where it is read. So Part 6, local, at the interface, sits opposite. Part 11, systemic, whole organism. Every other test in the battery is one more axis of that same split. Irritation, cytotoxicity, degradation, products, each a locus of harm named and isolated. This section explains how the body reduces to a single compositional pattern and why that pattern matters twice for a biomaterials reader. Start with 4 alphabets: amino acids, nucleotides, saccharides, and lipids. Each polymerizes into one of 4 macromolecule families, and those families stack upward from cell to tissue to organ to organism. The body is that composition, nothing more. It is a hierarchy generated by 2 choices only: which monomer and which linkage rule only, which monomer and which linkage rule. The real move of the chapter for a biomaterials text is dual use. Every one of these molecules is 2 things at once. It is the milieu an implant meets on contact, and it is a candidate material in its own right. So the body's parts list is a prerequisite twice over. It fixes what biocompatibility must satisfy, and it supplies the palette you build from. Learn the monomers, and the rest of the book's material choices become predictable. This part explains why ethylene oxide sterilization leaves a residue you have to remove. The same reactivity that sterilizes is the one you must then evict. Ethylene oxide kills by alkylation. It attacks the membrane, then the DNA, and the attack is blind to whether the cell is a contaminating microbe or the patient's own tissue. The gas that clears the device does not distinguish targets. What it did to the pathogen, it will do to the host. Porous polymers trap it, and it lingers as sorbed residue. Hence, a threshold, 250 parts per million in an implanted device, below which that residue must fall before the device is safe. ISO 10993-7 is item 7 of the biocompatibility series precisely because efficacy and toxicity here are one chemistry. This part explains how the body is built from a very small set of molecular building blocks and why that same list matters twice over for anything you implant into it. 4. Monomer alphabets, amino acids, nucleotides, sugars, and lipids polymerize into proteins, polynucleotides, polysaccharides, and membranes. Those in turn stack upward into cells, into tissues, into the whole organism. The body is a strictly hierarchical polymer economy built over a tiny inventory. The chapter earns its place in the book's arc by reading that inventory twice. Every one of these molecules is what an implant touches on contact, and several of them are what an implant can be made of. Interface and feedstock are the same list. Learn the body's building blocks, and you learn two things at once: the chemistry. A biomaterial must survive, and the natural biomaterials already on the shelf: collagen, elastin, and hyaluronic acid. This part explains how reference materials give a biological test, its scale. A biological test emits a number, and a number without a scale certifies nothing. Reference materials are that scale, a known inert control that should provoke no response and a known toxic control that reliably does run beside the test article. So that when you say this material passed, it means passed relative to a validated anchor. ISO 10993, Part 8 is the part of the standard governing how those anchors get chosen and qualified. That is what makes the verdict biocompatible, commensurable across labs, rather than a private judgment each one reaches by its own reckoning. This is the same move as a ruler meeting a defined meter. The reading means something only modulo, a shared reference. You calibrate the instrument before you trust what it tells you. This section explains why a biological safety test always rests on a trusted reference material. A biological test never measures safety in the absolute. It measures a difference from something whose response you already trust. The reference material is that fixed point, the material of known biological behavior against which the unknown is read. Qualify it well, and every downstream verdict inherits its credibility. Omit it, and a clean result becomes indistinguishable from a dead assay, because you cannot tell whether the sample provokes nothing or the test itself provokes nothing. The chapter's whole work is selecting and certifying that anchor. This is calibration, in the strict sense, the same move as a spectroscopic blank or a positive control. An assay converts to meaning only through a standard, and here the standard is a physical material rather than a number. This section explains ISO 10993, Part 9, the framework for identifying and quantifying potential degradation products. Part 9 is an abstract contract rather than a test. It fixes how you would identify and count whatever a biomaterial breaks into before any material-specific method exists. The concrete parts are its implementations, Part 13 for polymers, Part 14 for ceramics, Part 15 for metals. Part 9 is the interface they all satisfy. The ordering that everything else depends on is a precedence chain. You cannot ask whether something is toxic until you enumerate the products, and you cannot dose-relate toxicity until you quantify them. Identification precedes quantification, and quantification precedes toxicology. Degradation is treated as a stream to characterize rather than a defect to certify absent. The framework exists because a biomaterial is meant to respond rather than to stay inert. This section explains what Part 9 of the Biocompatibility Evaluation series does. Start with the object itself. A biomaterial inside a body behaves as a slow source term. It leaks its own decomposition into a living reactor, and those leachables decide biocompatibility. The bulk material does not. Part 9 is a framework rather than an assay. Its job is to decide whether degradation must be characterized at all, and to structure that decision before the material-specific parts, polymers, ceramics, and metals actually quantify what comes off. The move is to split one question into two gates, so screening precedes measurement. The first gate asks, does it degrade under use conditions? The second gate asks, what does it release, and is that dose toxic? Everything downstream in the series is this router, dispatching to a material's chemistry. This section explains the two ways a material can harm the tissue it touches, and the single structural axis that separates them. Memory. Irritation is memoryless. It is a local inflammatory response scaling with dose and contact, innate and immediate, resetting once the insult clears. You test it by measuring the reaction to direct exposure. Sensitization is hysteretic. The immune system learns the material on first contact, so re-exposure triggers a disproportionate allergic cascade below the original threshold. You test it by sensitizing first, then challenging. The standard ISO 10993, Part 10, pairs the two, because together they exhaust the local effect axis. Reaction without memory and reaction with memory. From this, you can predict the rest. Anything that reacts the same on the 100th exposure as on the first is irritation. Anything that gets worse is sensitization. This section explains how a material's local hostility splits along a single axis. Memory. Irritation is the innate memoryless response. It is a dose-dependent inflammatory flare. The tissue mounts on first contact with a leachable, with no prior exposure required. Sensitization is the adaptive one. It is a delayed type 4 hypersensitivity where an initial exposure primes T cells and only a later re-exposure triggers the reaction, now decoupled from dose. So the chapter tests two immune subsystems, not two severities. The reflex that reacts immediately and forgets, versus the one that learns first and strikes on re-encounter. Predict the assay designed from this. Irritation reads acute local scoring. Sensitization needs an induction phase and then a challenge, because you cannot detect a memory you have not yet written. This section explains why systemic toxicity is one axis of the whole biocompatibility battery, and how that single axis regenerates the rest of the list. A material is never toxic in itself. It is toxic somewhere, and the whole biocompatibility battery of Chapter 11 partitions by where the injury lands relative to where the device touches the body. Systemic toxicity is the far-field case. A leachable dissolves out, enters the blood, and damages an organ remote from the contact site. the harm travels, where cytotoxicity and irritation stay local. Read that one axis, and you regenerate the rest of the list. Acute versus subacute versus chronic is the same distant harm question indexed by exposure time. Pyrogenicity is its fever-shaped special case. The primitive is this: does the absorbed dose reach a distant organ? The ISO 11 test is merely the assay built to force that answer. This section explains how systemic toxicity sits among the safety axes of an implant. Safety of an implant decomposes into orthogonal hazard axes, one standardized test for each. Systemic toxicity owns the axis where the insult travels. A material need not fail at the implant site to fail the host. Leachables and degradation products enter circulation and strike organs far from the wound. So ISO 993, Part 11, asks what the blood carries elsewhere, the same substance dosed to the whole body rather than to one tissue. That partition is what makes the neighboring axis predictable. Local effects after implantation is the identical insult with its spatial reach set to 0. Chronic and subchronic toxicity is the identical axis with its clock run longer. This section explains how the 12th part of the ISO 10993 series fixes the input to any biological test. Before a biological test can mean anything, its input must be fixed. Part 12 of the ISO 10993 series is that fixing. It standardizes how a sample is extracted from a device, the solvent, the surface area, to volume ratio, the temperature, and the duration. It also specifies the reference materials, the known response controls against which any duration. This is a metrology move rather than a biology. 1. It converts a vague question, is this material toxic, into a comparable measurement by pinning the independent variable before the assay ever runs. A reference material is a ruler of known length. Without it, two lab cytotoxicity numbers are not the same quantity. Every downstream test in the series, cytotoxicity, irritation, sensitization, inherits its cross-lab comparability from this single standardizing layer. This section explains what Part 12 of the biocompatibility standard does. Every biocompatibility test above it measures nothing until two things are fixed: what the biology is exposed to and what its response is compared against. Part 12 supplies both. It does not test. It standardizes the stimulus and the baseline. You rarely challenge cells with the implant itself. You challenge them with its extract, leachables pulled into a defined vehicle at controlled surface area, temperature, and time. Read against reference materials, a negative control that should provoke nothing and a positive one that reliably kills. Fix the dose. Fix the 0. And only then does the claim that this material is toxic carry signal. This is the calibration move under the entire ISO 10993 battery. This section explains why a polymer implant's degradation products, rather than the solid itself, are the object of safety certification, and where Part 13 sits in the structure of the standard. The thing you certify is the set of degradation products. What hydrolysis and enzymes release into tissue over months is the real exposure. Safety is a property of the trajectory rather than of the pristine solid. You must name each fragment and measure how much of it appears. Part 13 is one slot in a factored assay. A shared framework, Part 9, is crossed with material class. Polymers sit here. Ceramics and metals sit as parts 14 and 15. The identified and quantified products feed downstream into toxicokinetics, part 16, and allowable limits, part 17. You can predict the neighboring slot from the axis, swap the chemistry of decay, and hold the identify, then quantify, then bound pipeline fixed. The standard factors along material by stage, and 13 is a single cell. This part explains how a polymer that will sit in a patient for years gets tested in months. You cannot watch a polymer rot. For the 5 years, it will sit in a patient, so you accelerate it. You raise the temperature. You force hydrolysis and oxidation in a simulated fluid until the same fragments appear in months. The wager is Arrhenius. Heat changes the rate of decay, not its chemistry, so the products caught fast are the products caught. Fast are the products caught fast are the products tissue would meet slowly. Then, the invariant pipeline runs on that concentrate, identify each fragment, quantify its mass. A real-time run sits alongside as the control, proving the shortcut invented no new species. Break the wager and the extrapolation collapses. That boundary is why both timelines are mandated. This section explains how safety gets judged for a ceramic implant. The safety of a ceramic implant is not read off the intact solid, it is read off what the solid sheds as it dissolves. ISO 10993, Part 14, performs a change of variables. It replaces the question, is this ceramic safe in the body, with a sharper pair of questions, which this ceramic safe in the body, with a sharper pair of questions, which species does it release, and out what the solid sheds as it dissolves. Identification fixes the what. Quantification fixes the how much. Those two coordinates are exactly what any dose-response toxicity judgment needs. This is one slot in the 10,993 series, which partitions biocompatibility by dissolution chemistry rather than device shape. Ceramics get Part 14. Metals and alloys get Part 15. Polymers get their own. Predict the pattern from there. Every material class earns a degradation product module because you assay the leachate product module because you assay the leachate product module because you assay the leachate. This part explains soft tissue at the level of function rather than structure. Soft tissue is a set of functions the extracellular matrix performs. Collagen holds, elastin springs back. Hyaluronic acid and chondroitin fill volume. Every device in the chapter is a stand-in for one of those functions. Sutures and tissue adhesives proxy the hold. Burn dressings and artificial skin proxy the hold. Burn dressings and artificial skin proxy the barrier. In Mendel fillers and breast implants proxy the volume. There is one design axis, temporary or permanent. Does healing take the function back so the material should resorb, as with catgut, fibrin glue, or a biodegradable scaffold? Or must the material carry it forever, as with PMMA microspheres and silicone? Fixing which extracellular matrix role a material proxies and whether that role is handed back predicts every material choice here. This section explains how degradation testing for metals and alloys is organized and why that concept sits where it does. A metal implant's danger lives in what leaks out of it. The body meets the ions and the particulate shed by corrosion, never the bulk solid, so safety is judged on the leachate rather than the block itself. The standard ISO 10993, Part 15, is the metals and alloys slot in a larger partition of degradation testing organized by release chemistry. Polymers, hydrolase, and that is Part 14. Ceramics dissolve. Metals corrode. Each pathway earns its own equantify and quantify protocol because the products of each watch-in and quantify protocol because the products of each differ in kind. Those two verbs are the whole content. First, identify which species emerge under simulated physiological conditions. Second, quantify how much of each, since toxicity is a matter of dose. From this, you can predict the siblings. Any material class whose failure releases something distinct gets its own numbered method. This section explains why a hard tissue implant is designed to match bone stiffness rather than to be as strong as possible. Bone is not a passive strut. It rebuilds itself to the load it actually carries, a principle called Wolff's Law. Because of this, the design criterion for a hard tissue implant inverts the naive one. The target is stiffness. Matching, the implant should match the bone's modulus of elasticity, and being merely strong is beside the point. Chapter 15's whole catalog obeys this single constraint. Plates, screws, hip stems, dental posts, every one of them is bound by the same rule. Match the bone's modulus of elasticity. Consider what happens when you overshoot it. The implant swallows the load, the bone senses disuse, and it resorbs. This is stress shielding, where the device weakens the very tissue it braces. From this one primitive, you can predict every move the chapter makes. Porous coatings invite ingrowth. Bioaerodibyl magnesium hands the load back as the bone heals. Stiff ceramic is reserved for teeth, the one place where a high modulus is genuinely wanted. This section explains toxicokinetic study design for degradation products and leachables. Every biocompatibility question splits in two directions: what the material does to the body and what the body does to the chemicals. The material sheds. This chapter is the second direction, toxicokinetics, the ADME fate of a leachable or degradation product, meaning its absorption, distribution, metabolism, and excretion. Traced after the other parts of the assessment have already named which species come out. The design releases a known quantity and follows it through blood and tissue over time. It is a mass balance tracer, not a harm assay. It answers where the compound goes, how fast it clears, and whether it accumulates. Toxicity tests read the effect. Toxicokinetics reads the exposure that drives it. You cannot judge a dose you have not followed. You reach for it only when a species persists or accumulates. This section explains why a shedding implant turns into a toxicology problem, and what changes once you frame it that way. A device implanted is a device that sheds. Its degradation, fragments, and leachables enter the surrounding tissue as an unintended, low-grade dose. Toxicokinetics asks the pharmacologist question of that dose: absorbed where, distributed how, metabolized into what, cleared how fast. Here is the generative move. Safety stops being a property of the material and becomes an integral of exposure over the body's clearance dynamics. The same fragment is harmless if it is flushed in hours, and toxic if it lodges in the liver or the bone for years. So the study design tracks fate and mass balance, and not intrinsic toxicity alone. You follow the substance through the body, and not the surface it left behind. This part explains what characterization does to a material, and why it comes before any question about harm. Before you can ask whether a material harms the body, you must fix what it is and what escapes it. That is the whole move of characterization. You convert a physical object into two specifications: its composition and its releasables, the fraction that can migrate into tissue. Everything downstream reduces to dose times. Hazard and characterization owns the dose axis. Chemical characterization names the constituents and quantifies leachables and extractables. Physicochemical, morphological, and topographical characterization fix structure, crystallinity, and surface, the geometry the body first toucher. The surprise is directional. A clean characterization can retire a biological test, since a substance that cannot leave the material cannot deliver a dose. This section explains what characterization means and why it sits where it does in the standard. Before a body can be tested against a material, the material itself must be fully specified. That is what characterization means, and its placement in the standard is the whole argument. The biological tests measure an output, the tissue's response. Characterization measures the input, the stimulus. What actually contacts tissue is never the bulk solid. It is the released fraction, the leachables and extractables presented across a particular surface chemistry and topography. Pin those down, and the downstream response becomes interpretable rather than a black box. Skip them, and a past cytotoxicity result tells you nothing about which substance passed. Same substance, different leaching profile, different biology. A material, for the body, is defined by what it gives off and how its surface presents. Its nominal identity is not what determines its biology. This section explains ISO 10993, Part 18, the chemical characterization of materials. Before you ask the body how it reacts, ask the material what it is. Part 18 is the chemical arm of the biocompatibility series. Its task is to identify and quantify every constituent of a device and, critically, everything that can migrate out of it, the extractables and the leachables, before any biological assay runs. The pattern here is substitution. A chemical inventory predicts toxicological risk more cheaply and more generally than the animal test. It front-runs, so characterization gates and narrows the biology rather than replacing it. Know the constituents, and hazard becomes a lookup against known toxicology instead of a fresh empirical question posed to a living organism each time. This section explains why chemical characterization comes first in the biocompatibility chain. Before you can bound what a material does to the body, you must know what the material is and what it can release. That is the whole of chemical characterization, the standard ISO 10993 Part 18. It sits first in the biocompatibility chain because biological risk factors as hazard times exposure, and characterization supplies the exposure term, the inventory of constituents, residues, and leachables that every downstream test, systemic toxicity, sensitization, then genotoxicity, tries to bound. Identify what can come out before asking what it does. A material whose extractables are fully known and benign needs less biological testing, not more. Characterization is the lever that shrinks the animal battery, working from outside it rather than counting as one test within it. This part explains what a scaffold is and what makes it distinctive within tissue engineering. A scaffold is the one component of tissue engineering designed to disappear. It is a temporary artificial extracellular matrix, a porous three-dimensional solid that holds a geometry for cells to attach to, migrate through, and colonize, while the other two components, the cells and the growth factors, do the building. Its defining property is timing rather than structure. It must resorb at exactly the rate the new tissue forms, the two curves crossing in harmony, as shown in figure 18.4. Read that inversely and the whole design space falls out. Porosity for traffic, pore size for occupancy, surface chemistry for adhesion, degradation rate for the handoff. A scaffold is a boundary condition with an expiry date. This section explains why every cell type used in a tissue-engineered construct is really just a coordinate in a two-axis trade-off space. Cells are the second ingredient of the construct, the scaffold's living cargo. Every named type you will encounter is a position along two gradients at once. The first axis is immunological distance from the donor site falls, a Y-east, or runs from autologous, meaning the cell comes from the patient's own body, to allogeneic, meaning it comes from another member of the same species, to xenogeneic, meaning it comes from another member of the same species. As you move along that axis, the risk of rejection and pathogen transmission rises, while the cost of harvesting from the donor site falls. The second axis is orthogonal to the first. It is potency, the count of how many fates a cell can still assume. It runs from totipotent to pluripotent to multipotent, narrowing at each step. Primary cells sit at zero potency, sorted only by their source. Stem cells trade differentiated function for developmental reach. Choosing a cell is navigating these two gradients at the same time, and every trade-off the chapter lists is predictable from where a cell sits in that space. This section explains the growth factor, the third leg of tissue engineering alongside cells and scaffold. A growth factor is a command. It is a soluble protein that tells a cell what to do next: divide, specialize, crawl, or stay alive. It does not contribute structure the way cells and scaffold do. If the scaffold is where a cell lives, the growth factor is what that cell should become. The step that does the work is this, meaning lives in the receptor rather than in the molecule. The same factor reads as proliferate to one lineage and as differentiate to another. And the families TGF-beta, BMP, FGF, VEGF, and IGF are defined by shared structure rather than shared structure. From here, you can predict the rest. Regeneration will demand not just placing these signals, but timing and localizing their delivery